All right. Well, look, I've got noon Eastern, and that's 1 p.m. Atlantic. I want to welcome everyone, thank everyone for joining. I have to tell you that we are incredibly proud to be speaking to you today in the midst of our first human clinical trial for our targeted hyperthermia therapy. Very exciting time for us. Looking back 2 years ago, we devised the strategy and runway for our targeted hyperthermia therapy and driving it to market. And it seemed like an extremely long road at the time. We'd identified that we needed to build a mountain of data around the safety and efficacy in order to be able to surpass the substantial hurdles from regulators. And we've done just a tremendous job at that. We'll get into a little bit of that. We've also been able to get here in an extraordinarily time and capital efficient manner. So these are things we're extremely proud about, and so keen to share with you some of the progress updates from the study today. Sona is developing its targeted hyperthermia therapy, which uses our proprietary and uniquely biocompatible technology to move the yard marker down the field of cancer therapies in the standard of care available to patients. And to continue that analogy, we're trying to get within field goal range. Now, it's still relatively early days here, and there's much work that needs to be done and much still that needs to be proven. But we believe that the therapy that we are developing with our device can be an immunomodulator in its own right. And for those of you perhaps not familiar with some of the immunology terminology, we'll dig into that. But what you need to know is that we are pushing really hard every day to develop for regulators a compelling package of evidence of its safety, tolerability, and efficacy so that we can get our therapy into the clinic as soon as possible. I'm David Regan, Chief Executive Officer of Sona Nanotech, and I have with me today my always indomitable and industrious partner, Dr. Carmen Giacomantonio, who is Chief Medical Officer of Sona. And we are delighted to welcome today a special guest panelist on this series of investor webinars, Dr. David Conrad, who is an experienced immunologist and clearly from his CV, a tremendous overachiever. I'm always daunted by those folks who have both a PhD and an MD, but beneficiary of it. Welcome, David.
Thank you very much. I'm happy to be here.
Thanks for joining. So first, of course, we'll be making a number of forward-looking statements today. So we refer you to our disclosure documents and to the risk of future unknowns that is identified in there. So today, the agenda, we're going to do a very brief overview recap on our cancer therapy. If you want more information, please refer to our website and some of our past investor webinars that delve deeply into the therapy itself. And of course, today we're happy to take your questions in this webinar. There should be a chat button on your screen. You can hit that and ask questions that I think are anonymous, and we'll endeavor to respond as best we can. So what makes Sona special? Sona has patented gold nanorod tumors. Here's an image of those gold nanorod tumors. That's from our most recent production batch. You can see the date of July 11th on there. Great consistency in those gold nanorods. But why are gold nanorods special? Why do we care about them? The advantage of them, they have a longitudinal aspect ratio, so they can be functional. They can be tuned to various wavelengths. They're uniquely biocompatible, used without particular toxin, CTAB, that is used by all others in order to make these. And it's been validated by the Nanotechnology Characterization Laboratory in the US, set up by the FDA and the National Cancer Institute. Uniquely biocompatible gold nanorods are ideal for applications of nanomedical practices in the body. This is what they're made for. This is the highest and best use for this product. Now, what is the problem we're trying to solve here? So Pembro, as it's called, also Keytruda, the #1 drug in the world, is an immunotherapy. It represented, and immunotherapies generally represented an enormous leap forward from chemotherapy and other harsher, shall we say, therapies. However, it has serious limitations. And as with all technologies, we're constantly pushing forward to do better. Unfortunately, to this study that's cited here, Pembro only works 21% of the time, depending on the cancer that's done. And what is needed is a therapy that can increase the response rates. In the low response rates here, Pembro responds, or Keytruda responds, when there is an immunologically hot tumor. So we need to be able to somehow create those hot tumors without inducing increased toxicity, which it happens if too much of this immunotherapy is applied. So I'm going to hand it over to Carmen to talk through how it is we achieve that.
Thanks, David. So we're going to just go, many of you, if not all of you, have seen this over and over. Just to remind everybody, essentially, this is the preclinical model that led us to our first clinical study. And the way it works is that we've used standard preclinical mouse models, and that's essentially the backbone of all immune research. We implant tumors into the mice that are tolerated by the mice. The tumors are actually, the cells for the tumors are developed from the mouse line. So when injected into the mouse, they're enabled to grow simply because the immune system is tolerant of them. And then we apply standard immunotherapies. And so the standard therapy that, well, in this case, what we're doing is we're applying our therapy. So we want to take that tumor that we know is cold because we've tried with several controls to see if the tumor responds, and it doesn't. And then we inject our gold nanorods directly into the tumor. We've gone over why directly into the tumor versus systemically. Directly into the tumor limits toxicity, increases accuracy and concentration of the gold nanorods, which is exactly what we need to optimize the amount of change we can make. And then when it's in there, we put a little probe in there. That little thermal coupling probe is going to tell us what the center of the tumor is doing, how hot it is. And the temperature we're trying to achieve, of course, is not to ablate it. We don't want to cook the tumor to death. We want to stress the tumor to death. And so then we apply, after that, we apply near infrared light. And this near infrared light, 860 nanometers, is passed through the tumor. And essentially, the gold nanorods are then excited. And the way it works is those little pill-shaped nanorods that you saw on the previous slide are excited to vibrate because of the way the electrons line up on them. And so that vibrational energy, as you can imagine, it's loosely speaking a way that heat is generated through the vibration of the friction of the energy imparted by the gold nanorods. That heat, fever, as we've talked about in many talks, fever, we're exploiting fever. That extreme fever heat, somewhere between 42 and 48 degrees, so 45 for the average, is a trigger for all kinds of things. One thing it triggers is the immune system. The innate system is awakened. When we get a fever, we feel poor because our immune system has been awakened and lots of things are happening. In this mouse model, immunity is being awakened. The second thing is these cancer cells that are so-called immortal, meaning they're immortal. The mechanism that makes them die is broken, and so cancer is essentially, that is the essence of cancer. So these immortal cells are not immortal, and they're not like super cells. They are fragile. And so the stress of heat will trigger a death cycle we call apoptotic cell death. That's the biological description. And those cells, as they're dying, our immune system clears up that process. And so cells called dendritic cells or antigen-presenting cells or phagocytic cells, so all of those things are kind of synonymous in saying these cells, your innate system comes in, consumes these things, and as it consumes anything in our body, as your innate system consumes it, the information, the protein in those cells are presented in a way that your immune system can see it, and the immune system decides lots of things from that. How much more immunity should follow? Is this a threat? Is it something that would activate a lasting and memorable response, or is it something that just needs to be cleaned up like a wound or an ulcer on the skin? So it's a very complex story beyond this, but essentially what we're doing is we're capitalizing on a very natural process by driving moderate heat into the core of the tumor to change the microenvironment, which is a big buzzword that is very critical to immunotherapy. So we've shown some great evidence here, and so what we're excited about is, the first thing as a researcher you'll hear, and Dr. Conrad's online, and I'm going to ask him to speak to this. But if you've treated a cancer in a mouse model and you cured it, the question is, is it idiosyncratic? Is it something just specific to the mouse? We have now treated 3 cold tumors, 3 different cell lines: colorectal, triple negative breast, and melanoma. Now, in a human, triple negative breast is actually immunogenic, but in these cancer models, these are cold. Cold means the immune system doesn't see it, and the implication is that there's not enough message, there's not enough antigen. Antigen equals protein. There's not enough message in that tumor for the immune system to be strongly activated. There may be some activation, but not enough. And so we've shown in these models that when we treat, first we grow the tumors, and then we treat with things that we believe should treat the tumors, like an immune therapy, it doesn't work. Or a control, like sterile water, it doesn't work. But then when we add our targeted hyperthermic therapy, when we actually stress that tumor microenvironment, we see great response. And so we've seen it in these models. And so my thinking, and I'm going to ask Dr. Conrad to comment on this, my thinking is that if you see this across 3 different cancers, and this is 2 different kinds of mouse models, that this intuitively would be translatable to the human situation, or at least more likely to be translatable. Dr. Conrad, I wonder if you can share your thoughts on, am I overreaching here?
No, I think you're making a very logical conclusion. The mouse models, I believe you said are blacks. Are they what are the mouse models? The ones?
Black 6, so that's either, and bulb C. So one is kind of there are 2 different kind of arms of the immune system, as we know it as in immunology.
Yeah, I'm very familiar with these. I worked with these in grad school many years ago. So the Black 6, this is a mouse model that tends to have more of an inflammatory, cell-mediated immune response to fight a cancer, to fight a virus. Valve C is more of a humoral antibody, less inflammatory. So one of the arguments against some mouse model work is that you can pick the mouse strain that tells the story you want to tell. You've picked 2 mouse strains that should tell different stories based on their immune backbone, but they're telling the same story. So I would agree that the fact that you're getting similar results in seemingly immunologically disparate, genetically disparate mouse models is really supportive of this being a generalizable trend, a generalizable mechanism. And having it work so well in multiple different and cold cancer models, I don't know what more you could ask for in a mouse model to suggest to take it to the next level.
Excellent, thank you. Yeah, so this is the excitement, and I'm very fortunate to be at Dow with scientists like Dr. Conrad and other people who have looked at this, and thankfully during our inter-lab and lab meetings and our presentations at rounds, we get the edgy questions that really go to the core of, is this real, and what are we seeing, and what are we missing? So, but we're very excited, and again, this is the stepping stone that made us confident we should be looking at this in the clinical.
Mm-hmm. Mm-hmm. And that's why when we get questions, when folks say, look, this is great, but it's mice, what do they matter? But triangulating it with different models gives us the confidence and the optimism that this can translate to human. And, now we're in this human trial, we will see. So let's jump in and give you some updates on where things are going there. So we're going to talk on objectives, the status, and also importantly, success factors. So first and foremost, we are doing this clinical trial. It's an early feasibility study trial, which is always the first study for a medical device. We're doing it in Chile at a clinic called Bradford Hill. Now, why Chile? Chile has a high incidence of melanoma. It has fast regulatory approvals, and importantly, it has immunotherapy as standard of care in the public health system, which ought to help us to enroll faster. Now, that's the clinic on the left, and you can see on the right is Dr. Carlos Rojas, our principal investigator. Here he is presenting at the American Society of Clinical Oncology in 2024. I've got a Merck logo up here because that's one of their biggest clients. It's a highly reputed clinic, great jurisdiction to be doing trials of this sort. So we're very fortunate to be working with Dr. Rojas and his extremely capable and well-staffed team. It's a leading cancer clinic. It's been around for 15 years, and they focus on immunotherapy, which just makes it perfect for us. So, Carmen, do you want to speak to the objectives for this study?
So as David said, this is an early feasibility study, and as for a device, what that translates to is the key objectives are, is it safe? So in other words, we've shown this in the lab. We're very comfortable, hundreds of applications of this. Translating it to a human, there are obviously other factors: the human condition, the amount of pain or discomfort they might receive, etc. The amount of anxiety and the willingness to subject to it. So evaluating the safety of this, both from the application in the clinic, from the patient experience, and from the provider or the healthcare provider's experience. We're using a laser, and so laser has connotations of risk. And so showing that this laser at 860 nanometers, which is the short end or the near end of the laser spectrum, it's far better tolerated, far safer in the environment. And because of that, and of course, we've had to go through all the safety regulations that use this in the clinic. This is a low risk situation but that's the first objective. We can say that we're proving that. The second thing is tolerability. And again, so we're talking about tolerability. We're talking about there's a lot of factors that go to tolerability, not just the patient tolerating it in the moment. The patient, it's all very exciting. It takes minutes to an hour, depending on how many lesions, how many tumors we're treating. But the tolerability is what happens after that as well. When the freezing comes out and the patients are in their own space, is it still tolerable? Is the response still tolerable? The changes in the skin? And the other tolerability is from the provider. For this to translate into a clinical experience, the person applying the therapy has to be able to do so reliably, repeatedly, and comfortably. So, because again, the more, this is translational research. We're taking it from a lab. Truly, this is bench to bedside. And so when you do that, some of the learnings are, how do you modify what the lab tech person is interested in? How do you transcribe and translate that to a simple device that a clinician and a technician can apply? As you know, a technician in a hospital doesn't always know all the intricacies of the machine they're working with, but they know all of the ability and inability and all the risk. And that's what we're translating right now. So this tolerability. And then once we get past that, we want to show, so this is the bullet 1 and bullet 2 are the focus of this trial. The third thing, though, and what we've talked about all to this point, is, are we achieving what we believe we're achieving? And have we shown some impact in that tumor microenvironment? Now, the setup of this kind of trial, being it's in South America, we don't have obviously access to fresh tissue right away to do the fancy lab things we're doing, but we have access to tissue. Tissue meaning we will be, and there will be slides to go over what the protocol is, but we are looking for before and after effects to give us the confidence, the signal that yes, this is activating an immune response. And so we're looking at that tumor in terms of taking it, just like if you went to a hospital here and you had a tissue biopsy done, pathologists are going to look at this and they're going to look at the tissue before and after the treatment. And we're going to see, is there some evidence that we have actually achieved an immune response, some reflection of immunity that we expect? We're very confident based on everything we know that that's happening. And this will give us our earliest insight in the human situation.
That's right. It's from a regulatory perspective, these are set up to look for safety and allow the device developer to guide the development, the modifications to their device that are necessary following some time in the clinic, in actual times. Of course, everyone looks for some efficacy data in there as well, but that's not what the regulators are thinking about.
But we are we are interested in that too. It's just we can't keep it right on it's not the horizon right now.
So on the trial overview and the protocol, if you will, for the entire trial here. So 10 patients is what we're looking for with late stage metastatic melanoma. who have not responded to the best therapies, the current standard of care that is available. And for those people, we're following a protocol that's a 28-day, guess I've got 30 days on here, 30-day protocol that entails biopsying them up front, taking a blood sample up front, assessing and characterizing the nature of the tumors before our therapy is applied. Day 1, we apply THT. We do it again on day 8, monitor those things. On day 15, take some samples, assess things, and at the end of the study, do another biopsy, take blood sample, and assess the progress at that point. That's a cycle of time. Of course, that's laddered over time as we have patients enrolling in. And so all the data does not come at once. It comes rolling in over time. Anything to comment on for that, Carmen, having been there in the coal face?
Yeah. Yeah, no. So this is, and you talk about laddered is the right term. There's a process for the clinic. So because this is all new, there's a tremendous amount of personnel into this to make sure every step is being done, to record every step, see what's working, see what could be tweaked to work better. Day 1, so these tumors will have been biopsied. If they haven't been, we'll biopsy them on the first day of treatment. But these tumors will have been biopsied, or at least a representative tumor. We need at least one for comparator. So we'll apply the THT, and then the patient will come. At the time of the first biopsy, we're photographing the tumors that are on the surface of the skin, or we're measuring with ultrasound or other parameters the tumors that are deep to the skin. Obviously, if we know they're there because almost all the tumors, the vast majority, will be palpable. We can feel them under the skin, but in order to measure them, we need so that adds an extra critical step of knowing where they started and where they finished. Where this trial isn't expected to show dramatic changes in size because, as you pointed out, it's 30 days. But any change in size will be important for us. And we know that in immunotherapy, we can actually see progression. We call it pseudogrowth. If there's a tumor with volume X and it is infiltrated with immune cells, we might see on day 8 that the tumor looks bigger than the day we started. And that isn't tumor growth. And in the world of immunology, we've learned that now in the clinic, that early on, tumors swell up with inflammation. And the inflammation is the immune system and the inflammatory cells doing their thing. And so, but we don't know what's going to happen in this under the influence of our therapy. So that's part of what we're doing here. And so it's going to be very careful. And we're going to have to be very careful when we look at that and measure it in the context of that histology, of looking at what's really going on inside the tumor or under the hood of you.
Well, we talk about our THT, our targeted hyperthermia therapy, as being an immunomodulator. So what success is to us here is the seeing that is actually modulating the immune environment.
Exactly.
That's what matters more than anything else. So this is our protocol. Carmen's just come back from applying it, from training. We're going to go a little show and tell now. We're enrolling. We're happy with enrollment. Things are coming along, and we want to give you a little flavor here of what was going on down there.
Yeah, so this is us. This is the Fantastic Four, getting ready to treat the patients, and me on the left, of course. Barry Kennedy is a technician, scientist that works with me closely in the lab. Len Pagliaro, many of you will have seen on webinars. Len is actually one of the founding fathers of all of this, and very excited to be there. It's been really a life's passion for him, and he was quite excited to see his dream finally come true. And there's Dr. Francisco. I won't attempt to say his last name. I'll insult him. That's an odd thing to say for Giacomo Antonio. But this Francisco is an interventional radiologist, a very keen guy, and he spends half of his professional life in this clinic working on these novel inventive trials. So this is us in the clinic, day 1. We gown up and set up. The clinic is all done as a sterile situation as possible. And we're having talks. This is literally first day, first hour, talking about the logistics of setting up. You can see around Francisco's neck, we provide safety goggles to everybody in the event of stray laser that could be reflected off of a shiny object. Then we're setting up, of course, and this is you can see. So this is part of the info. Why are we doing a feasibility study? Part of this still looks like a laboratory. That's a ring stand that Dr. Conrad will know, remember fondly, I'm sure. And so we're holding, this will translate to a device that has a fancy arm, an appropriately balanced arm that will hold the device appropriately over the patient in time. And so our engineers will look at this and look at the logistics and then look at the patients. And the next slide. So then we'll see what that should translate to. So what you're seeing here is I'm standing in the background, Barry's standing in the background. The doctors, the nurses, and the technicians are doing the work. The other thing about a feasibility study is it has to be feasible, it has to be translatable. And so it is important that the team at Bradford Hill administer this therapy. We are there for moral and directional support. As in all, if you've ever been in an OR, any of you, and I've been in lots of them, where the industry technologist is there with us to help us to make sure we apply their technology appropriately. You can't interpret the results if it wasn't applied appropriately. And then there you can see the patient is already in the room getting ready to be treated. And of course, so this is the gold, and the gold doesn't look gold; it looks purple, and it's just the way the gold refracts the light that gives that appearance. And these are the syringes that we would be using, or in fact did use to inject the gold into the patient. And the patient is lying on the gurney there, getting ready. And he has been draped and prepped. Next.
And that was a really extraordinary moment for us, the first time injecting gold nanorods into a human, and then doing it at this clinical trial in Chile.
That was actually quite a monumental event, just to see the first injection go in. Now, that we would be far more emotionally attached to that, I'm sure, than any of the viewers today. But I can tell you get chills up your spine when you see the first injection, for sure.
That's it.
And then, so this is the application. These are two different tumors. One on the left, you can see that is our device. The tube over the top is the laser that's supported, so it's immobilized in that device. The little wire coming out of that's a skin-based lesion. You can see there's a pink lesion on top of the skin, and overlying that is a coat of aloe vera gel that helps protect the skin from being burned by the laser energy. It doesn't burn. The energy, if you apply it long enough, will cause the skin to burn for sure. This to the left is a camera. You can see that's one of the techs are holding the camera, and that's constantly monitoring skin temperature to keep it under 50 degrees, so that if it goes above 50, regardless of what the tumor heat is, the machine is turned off and things are either repositioned or we have to rethink the treatment so that we're minimizing chance of inadvertent injury to surrounding tissue. On the right-hand side, what you see is this is actually a very common situation. That's melanoma. You can see some staining. That's melanoma staining in the skin, which is common when you have a melanoma cancer tumor in the soft tissue beneath the skin. And there's two injection sites there where Francisco has injected the tumor deeply into, not the skin, but deeply into the tumor. There's three actually injection sites in multiple directions to infiltrate widely. After this is done, we wait 2 hours, and the 2 hours is to allow the gold to permeate the tumor more widely so that as the energy is passed through the tumor, the energy comes down. So it's not a pin or an arrow. It actually comes in a cone, comes down in a cone. So as it transports through the skin, there's a wider area of about the same dimension as the tumor. So there's a metric that we use. There's a table that says if the tumor is 1.5 centimeters wide, hold the machine so far away from the tumor so that the cone will essentially activate the gold as it encounters it. And then the energy that's passed through, it's always 860 nanometers. Then we can vary the amount of energy, the milliamperage that we use to do that from that point. So that's the technical, as far as I'll go with the technical, but you can see you can imagine there are different challenges with a subcutaneous, the one on the right, lesion versus one just sitting right up there on the skin where you can stick that little probe in there and jump. So they're the easy ones. And that's actually more of what we see in our clinics. This is actually what motivated us to design a lot of the device. And that deeper one is a challenge that this trial is helping us to solve. How best to get to those deep tumors and spare the skin. And these are some of the learnings that we're thankful for the clinic and the people Don't forget, these people are subjecting themselves to this. We're not promising a cure for they're contributing to the knowledge base of cancer. And so we're eternally thankful to cancer patients that are willing to, in all trials, all of my life, I've experienced these people, and I'm more than thankful. Yeah.
Carmen, there's a question here about how many tumors per patient we can treat under the protocol, or we do treat.
So we treat up to 4 at this point. That is an arbitrary number, and it's for the purposes of the trial, because we didn't know and still don't know what the additive length of time is. So if a superficial tumor, that one on the left takes literally 10 minutes to treat, start to finish, once the patient is in the room, etc. Put the probe in, the gold has been in for 2 hours. So then once we activate it, it takes 2 to 3 minutes to get the tumor up to heat. And then we want 5 minutes of that heat at 45 plus or minus degrees. On the right-hand side, there's some work you have to do to balance tumor heat from skin trauma. And so that takes longer, and that can take up to an hour. So 4 lesions could take 4 hours. Now, we've treated, our third patient will be completing treatment tomorrow. The longest we've spent is 2.5 hours in this situation. So and that's with 4 tumors. So we think that this is so again, feeds into tolerability. How long can you expect a person to lie on a gurney and receive a treatment of any type? And how long can you expect a team of treaters to administer the treatment? When somebody receives treatment for cancer, so when somebody has a PET scan, which we do often, it takes 3.5 hours to get that done. Plus the time it takes for the stuff to circulate. So we are familiar in healthcare with long timelines for testing. We are, for sure, this is part of the feasibility purposes of the study, to figure out our timelines.
And Dr. Conrad, you spoke of with the mouse models and the fact that they're genetically disparate. Is that an important factor when you talk about a human trial in terms of the population base?
Yeah, I think if this group only focused on one mouse model, and honestly, if I was picking a mouse model, that I would pick the one that favored the response that I wanted to see. You could rig the system to make results look better than they otherwise would. So again, my point before was you're using mouse models that, if it was purely based on the genetic backbone of the mouse, so the characteristics of the mouse, the tendencies of the mouse's immune system, you really shouldn't be seeing it work so well in both models, because the elements of the immune system of both models are so different. So the fact that it works so well really speaks to a more generalizable mechanism that's going on. And to talk about extrapolating that to the general population, I mean, I've got identical twin boys. Outside of identical twins, everybody is genetically diverse. So, I mean, that's a drop in the bucket, but it's better than most mouse or most animal studies that would focus really in on one model that works best for them. This is a little outside the box. It was, I think, honestly a bit risky to do it in 2 such disparate models, but fantastic to show such promising and positive results in both. And I mean, one other thing, anyone who's read about it or who's going to do some homework about it, one of the other criticisms of working in mouse models is, in mice, the tumors tend to be more immunogenic. So you tend to see better responses in a mouse model than you would in humans. But again, as Carmen mentioned before, these are cold models. These are cold cancers that don't trigger that immune response that, again, I would pick different ones to make it look better than it otherwise would. So it's like you've got this deck stacked against you from an immune system point of view, and it still looks really good. It's still showing that things are working the way you're hypothesizing. And to get these positive results, again, in these 2 very different, immunologically different models, I think is fantastic reason to expect good and similar results in humans, which by definition are also going to be very genetically and immunologically different as well.
Right. So it's also then a game of numbers, because we only have 10 in this case. So
Yeah.
so that is limited. But in terms of stacking the deck against ourselves, we've also got limited tumor size here, a limited number of tumors to be able to assess. And we're doing it, of course, what I mean to say is in late stage melanoma, for which every other application of standard of care has not worked. And yet we're going in trying to make it work. So, I don't think of it so much as stacking the deck against ourselves as giving us the toughest challenge and show that we can make it work in that environment. That will make the path forward easier and clearer.
Oh, absolutely. The
Okay.
newer treatments are largely immune based. We're not coming up with newer and better chemos so much these days. The focus is on the immunological because running out of other options for these folks.
A couple other questions here. Question: activating the immune cells versus if the immune cells are killing the cancer cells. Can you tell if apoptosis is happening in these photos?
So it's a great question, and the answer is you can't tell from the photos. Certainly not the early photos. These are just early treatment. You can even if the tumors shrink, you can only surmise what the pathway is. We need tissue to do that. The only tissue, as again a function of the study, that's not set up for a lot of tissue analysis because of the nature of this first feasibility study. We will have, and we're thankful to have, some blood work, and we're thankful to have some histology. And the histology will tell us, and I'm going to show you a slide about that. So this is a very good question because we have a slide that helps us to understand the difference between an immune cell response and an effective immune cell response.
Right.
So we'll wait till we get to that slide to finish that.
Okay, we'll fast forward. Here we go.
So here we are doing more treatments, and this is Francisco, and this, again, speaking to the point, there's a technologist holding the camera, monitoring the skin. You can see on the left, the therapy is being set up to be applied, and then Francisco himself is operating the machinery. This is not us doing the treatment, and that's very important for everybody to know that this is the transferability of the therapy. This is our first or second patient, I can't remember which one, but this is already that person, this Dr. Francisco is on the curve. Yeah.
And you can see the fiber here, the laser fiber is actually incredibly thin relative to the handle on the end that's used, again, just for functional purposes. That, as we've always said, opens the door to being able to access many different points, deeper in the body with such a thin fiber.
Yeah.
We have a question here. Do you expect better immune response by stressing the tumor rather than boiling it?
Yes. Great question. That's the right question. So you want to, if we boiled it in my clinic in the current world, we freeze tumors, cryotherapy. We heat tumors with microwave therapy. We heat tumors with electrodesiccation therapy. If you ablate a tumor, there is a good chance there will be some protein left over, and the immune system, no matter how the death occurs, the immune system, the innate system, comes in and essentially, to simplify it, eats up all the debris and presents whatever is viable to the immune system, or not viable, but presentable. The better if the cell death, if the tumor death or the tumor cell death that you initiate is part of an active process, that's apoptosis, then there's likely, and it's just a logical assumption, more protein, and the process is far more, so there's more protein available, more viable antigen. In a cold tumor, it's called cold because if a hot tumor has 100 proteins and they're all visible to the system, that's hot. If a cold tumor has 100 proteins and only 5 of them are visible. So if you're going to ablate that cold tumor, there's a good chance, relatively speaking, you're going to lose a lot of that information you need. So if you dismantle the tumor through a natural process that your system consumes and presents, there is the hypothesis. Our hypothesis is the likelihood is that you'll see better antigen presentation. We are proving that in the lab. The next iteration of publications we have coming next month will show you that we believe we're proving that. But for now, that's the hypothesis. And so a lot of clinical work is a very good, it's a ladder of effect, as David said. So you have a hypothesis, you move on the hypothesis, you learn something, you generate new hypotheses. Sometimes in the process of that, you make conclusive findings that so that you've proven your hypothesis. Now you have a known effect. And so this is what we're evolving through. We are literally at day one.
Excellent. So let's go to the next slide here, which really gets into what are the success measures with this study and what are we going to be looking for here when we do have results, and our timing on that, is, as I say, enrollment is going well, and we're still optimistic that we hit the target we've set for the end of the summer.
Yeah, so success is just, to reiterate, we delivered the therapy as we described. It will be modified along the way as the patients and the system teaches us, but for the most part, with minimal adverse events. What's an adverse event? A patient couldn't tolerate it because it was so painful, or a patient has a severe reaction that limits their ability to come back for more treatment. That's a severe event. A moderate adverse event would be skin irritation or even a skin burn. That you can, that's why we put the aloe on. And so for those deep tumors, we're learning, just for logistics, we may have to get past the skin with an alternate device to make it more accessible and to really focus the energy on the tumor. Tolerability, again, both from the patient side and from the system side. Is this something a clinic would adopt? And it makes sense from a feasibility. Is it feasible for clinics to adopt this and treat patients en masse? Or is it just so much that this can only be adopted in larger hospitals with far more resource to support it? So we're trying to finesse this and modify it so that it's applicable in most clinics, as Bradford Hill is. And then the tumor microenvironment, and this is by its very design, the third objective, and flip it on its head, the grail. We want to see that change in tumor microenvironment, and this study will be allowed to do that through histology. To give you an example of what histology is, I'll show you in a second, but we are going to take patient samples of tissue and stain them and look for before and after changes. So, and then, the learnings developed. Yeah, so this go on. So success is all these learnings. The next iteration, our next trial that we'll do here in Canada, will have all of these learnings embedded into it, which make it a far smoother experience for the clinician and the patient, and a far more useful than experience for us as clinician scientists to understand the real biology of what we're doing and to know where next to tweak it. We are, when we have a good hypothesis, a very sound hypothesis, and Dr. Conrad, I believe, agrees. We don't know what we don't know. That's what trials are all about, is learning how to do it better and optimizing your hypothesis.
Yeah, that's it.
So this other thing, you see, you have something there potentially to treat other cancers. So as we've been doing this, these surface lesions, the most common cancer, or the second most common, depending on what statistics, is squamous cell cancer, not melanoma. Melanoma is the most deadly. Squamous cell cancer is the most common. And it's a real challenge for people because it grows with such vigor and such frequency. And so the surgical management is very challenging. And for some people, for a small percentage of squamous cell cancer, it's extremely dangerous, if not lethal. Transplant patients have an extreme amount of squamous cell cancer. So our next cancer will likely be squamous cell because it's the natural next target for us. And this, as you talk about laddering, as we're rolling out melanoma, we'll be initiating squamous cell.
So we're getting asked here if there are any adverse events to dates or intolerability or how many patients we've dosed, and it's it's not appropriate to comment on that in the midst of the study. Too early to be talking about this stuff at all. But high level, Carmen, to put you on the spot, level of optimism or overall commentary on progress of the being in a study, being in the first human study overall.
I think it's fair, and people would understand these are humans, so we can't really divulge on a patient-to-patient basis what's going on. That would be inappropriate. A lot of this stuff, because we're not the providers. The providers are, as I just introduced, our Francisco, his nurse, and the other team. The follow-ups will actually be done by them, and we won't be part of it. And we're not allowed to personally assess the responses. We can only see it at the end of the trial when they present us with their findings. It's because clearly I have a different eye. Things I see it differently. And so, yeah, so that's enough to say. But am I excited about it? The learnings, this clinic is spectacular, and the learnings and the opportunities to see what the real challenges are in the human setting. It's, you know, good and bad. When this is all done, we'll sit down and we'll have a, you know, coffee, and I'll tell you all about it. But this is exactly what we need to know. So I am over the moon excited to be in this
Yeah,
situation.
couldn't be happier with the choice of clinic in this case. So now this next slide is we're going to get Dr. Conrad to comment on too, because it gets into the specifics of what we're going to be looking at when we come back with results. Hopefully towards the end of the summer. Can you take us through this? It's a little complicated, but I think you can break it down between the both of you guys.
Yeah, so I'll briefly. So this is essentially an example of what we will see from this trial from histology, so pure biology. And there are 4 panels here. On the left, this is control. This is an untreated tumor. And then on the right, the next panel, panel 2, is what would happen if you treat something with an immunotherapy, in this case, interleukin-2, and the immune therapy is ineffective. And what you're seeing there is it's all clustered at the edge of the tumor. It actually doesn't penetrate into the tumor. So the tumor to the right is free of the effect that we're trying to achieve. And we see this in cancers very often in the human setting, that person on immunotherapy has immune cells at the edge of the tumor, but they're not getting in. And that's that tumor microenvironment. And that's why the tumor microenvironment is such a big focus of this device and this effort, is to change that microenvironment to enable cells to infiltrate. Those cells with the arrows on the edge and panel 2 are literally knocking at the door. In panel 3, this is what targeted hyperthermia does. It changes that so that the cells have now infiltrated into the tumor. Now, when you look at that, you say, why isn't that just riddled with cells? Well, this is the effect of THT alone. This is an immune infiltrate of natural. This is a natural response to immune infiltrate. And it only takes a small amount of immune cells to affect a massive change, because these cells are not individually. There's enzymes being released from these cells that are causing these cancer cells to die. And so that immunogenic cell death, or that immune cell death, is the effect. A small army achieves a big result. And then, of course, to the right, the 4th panel, what we're seeing is if you take those immune cells and then you fire them up with a Keytruda or a IL-2 or whatever immunotherapy is on your shelf, you can invigorate those cells to recruit more cells into the fight and hopefully have a bigger effect. And so this is exactly what our immune modulation is. We're focused on we're looking at panel 1 and focused on making panel 2 turn into panel 3.
And so really it's the proliferation here that we're looking for, right? How widespread those brown cells effectively are.
Dr. Conrad, I'd be interested in your thoughts on what
Yeah.
I just, as a pure scientist and a pathologist.
Yeah, I mean, this is the stuff I look at pretty much every day. In the second panel, you can see there's a cluster of cells that are brown, and of course all the purple is just all the nuclei. That's the counter stain that shows where every cell is. And the slightly bigger, more pale cells are the tumor cells that are adjacent to that cluster of brown cells. So panel 2 shows a cluster of T cells, and that's just a little reactive collection of T cells. They're not really doing anything inside the tumor. But if you look at the 3rd panel, there's distribution of the T cells into the tumor tissue. So they're actively moving through it. Like there's no roadways, there's no blood vessels to take them there. They're actively infiltrating and moving throughout that tumor tissue, which it has to do to mount a response. And then the last panel over, just the difference is even more striking. There's many more T cells that have made their way into that tumor tissue compared to the control on the far left, which is pure tumor and really no T cells in there whatsoever. Yeah, so yeah, I agree. This is showing what we want our immune system to do to fight a cancer is it's being boosted by the THT, and even more so when THT is coupled with IL. Yeah.
Excellent. Okay, thank you for that. Now, moving on beyond this study, looking at next steps, a couple of things we want to touch on is the next study. As we say, this early feasibility study is to pave the pathway for the pilot study for a medical device, and there's some future developments to talk about too here. But in terms of the next study, being a pilot study, we're aiming to do that in Canada. And the goal here is a deeper dive into the treatment effect. What's going on? It's going to be a larger number with 30 to 40 patients. This is how the typical protocol rolls out. 10 patients in your first early feasibility study, and then 30 and 40 with more of a focus, again, on safety. But then from a regulatory perspective, then they do want to look for some efficacy data there too. We think this can be a highly efficient study because we think it can be largely under our control. We will not be outsourcing it to a so-called CRO, a clinical research organization that will charge you an arm and a leg to take that out and do it. It's potentially multi-center, which would be beneficial from an enrollment speed consideration, and we're targeting to initiate it by the winter of 2026. We're looking to roll very quickly from this study into the next one, and we think this study will give us the support we need to get permission to initiate that study. Now, this pilot study, of course, leads to a randomized clinical trial called, in the medical device terms, the pivotal trial. That's longer term, that's bigger, that's going to be 100 to 200 patients. But you have to earn your way to get there, and that's what we're doing. So from a perspective of the company's pipeline, and this slide will answer a couple of the questions that have popped up here. Carmen, why don't you handle this in terms of where we are? Across the top here, you see the stages here. Discovery and concept design moves towards preclinical studies, moves towards early feasibility studies. That's obviously where we are with melanoma, which then you graduate to a pilot study and a pivotal study. In terms of the prioritization of types of cancers, I'll let you speak to that, Carmen.
So we are clearly we're now in the clinical phase of melanoma research. What's become apparent to us from the melanoma research, both in our lab environment where these are very accessible lesions to the clinic, it's become crystal clear. Squamous cell cancer will be obviously the next. Now, squamous cell cancer is cancer of the skin, so cutaneous squamous cell cancer, but there's also squamous cell cancer of the mouth, the oropharyngeal cancers, and some other areas of the body. So these squamous cell cancers are ideal targets for what we're doing. The melanoma is, and this early feasibility work we're doing is honing the process of delivering the therapy. But clearly, these two cancers will be, again, laddered while melanoma is rolling out. And there's not, in fact, not a whole lot of very good clinical models or preclinical models in squamous cell. So the squamous cell clinical work will actually go quite quickly from left to right using the learnings from the melanoma, which are both cutaneous. So the biology isn't as important then as the anatomy, and the anatomy has been sorted out with the melanoma. Then breast cancer, and again, thankfully through this trial, we're learning about how to penetrate beneath the skin and spare skin trauma, and that will lead us quickly into breast cancer. And as some of you know, we have an immunotherapy breast cancer trial going on right now, an intratumoral immunotherapy breast cancer trial going on at our breast health center. And so this will be, again, having achieved the safety and the efficacy of introduction into humans, this will be a natural cancer for us to add to, so probably third in line. The colorectal, we have tremendous clinical data, preclinical data coming out in colorectal. And I did see flash on the chat box, when is this? So we have a colorectal study coming out next month, and we have a targeted hyperthermia, true biology, a deep dive into it coming out. And these will come out in slight sequence, but only a week or two apart. And these are the big mechanistic studies that we've been working so hard on the last year. And so colorectal is definitely on the list of things to treat. Colorectal is going to take some. We're going to need some technological development. We need to develop the device that's going to get us into the lumen, into the rectum. And so the engineers that will have helping us will help us to solve that problem. But that's definitely solvable. We do transrectal surgery all the time now for some of you that know about colon and rectal cancer. So we do surgery through the anal verge all the time. So we don't anticipate a whole lot of challenge aside from looking at devices in a different way for that. And then glioblastoma, we're excited about. We're just got our ethics approval to go into glioblastoma and here at Dalhousie. It's a glioblastoma is a brain tumor. Lethal.
Yeah, that's not for us, right?
That's sorry.
Going into Dalhousie, yeah, but not Sona.
No, not Sona,
Yeah.
but it's preclinical work that we're doing that Sona will be fed into Sona's pipeline. Bladder
Right.
cancer definitely for us, and bladder cancer is an immunogenic cancer. It's now treated with a thing called BCG, tuberculosis vaccine. We know how responsive it is to immunotherapy, so it's a natural cancer for us to target, and it's a surface cancer, even though it's in the surface of the bladder. As David pointed out earlier, this device was originally designed to actually pass through a scope. So we've already overcome the hurdle, the limitation of getting it into the bladder, because the device is actually naturally designed for passing through a scope, like a cystoscope, a scope that would look into a bladder. And then finally, this triconjugate stuff, and that's really bench top still, and we'll be scaling up though for translation and delivery into the clinics. And I'd let David talk to that timeline because it's more of a, there's a lot more moving parts to all that.
And this speaks to, there's a question around, can these gold nanorods carry other medication or compounds into the tumor? That's exactly what this triconjugate concept is about. That goes back to part of the functionality of gold nanorods and their ability to carry payloads. Opens up some really exciting concepts, and we've been filing a number of provisional patents in that area and doing some work in the background. Well, certainly focused on getting this trial off the ground. We see that as laying the groundwork for the future products, frankly. And then finally, we have, speaking of, similar concepts around a theranostic, the idea that using the gold nanorods and the theory, the principles of targeted hyperthermia therapy, that a product could effectively be developed to identify cancer and treat it coincidentally. Obviously, extremely early days there, but exciting in terms of the possibilities. And that's why we do see this technology and its core biocompatibility as being a platform technology, opening up many possibilities that we are really keen to pursue. Going to, in the interest of time here, going back one slide, sorry. And there we go. So timeline here. Challenging. A little delay on that mechanism. In any event, timeline, there was a question around whether we're talking about the winter January 2026 or December 2026. Certainly, we're talking about this coming winter in terms of for our pilot study first dosing, first half of 2026 is where I've identified that, and then aiming to have initial readouts in second half. We've got our initial readouts targeted here for Q3. And that is was indicated by the end of the summer. We also have, Carmen just mentioned, I do want to underline it. We're expecting to have 2 papers published under Carmen's leadership with our team and support. And this one in particular delving into the mechanism of action. And we expect that will create some attention within the industry who are really hungry to see, okay, these look like, great results you're putting out. Help us understand why those are happening. Dig down to the next level here and allow that. Carmen, any comments on that? Did I get that?
I think you've summarized it well. We really what we're looking at in this mechanism of action is we're applying the targeted hyperthermia at checkpoint A, and then we're sequentially analyzing the tumors to understand what the biological changes are over time and what happens at those particular times if we intervene with something else. And so we're trying to really understand the mechanisms of action, both of targeted hyperthermia and of how the immune system is. What this is doing as an enabler, an immune modulator, an enabler, is it's opening up opportunities to intervene at opportune times within the development or treatment of a cancer. And so we hope to have some very exciting insights into that.
Another question I want to hit you with here is, could nanomedicine realistically replace chemotherapy in the future, and what would need to happen to make this a safe and effective option? I think that's very much what we're pursuing now, but
yes, the answer to that is replace is a big word. It's certainly going to overtake chemotherapy. There are some, it's to get into the weeds, there are blood-borne diseases that are going to require systemic treatments, and there are solid tumor diseases that are slightly different in their approach and target. But no question, nanomedicines meaning these minuscule, targetable, precise devices and capsules of treatment are definitely the future. The biggest limitation in immunotherapy today is toxicity. As David said early on, immune therapy, the way it works all now is it requires that the immune system has engaged the tumor and then for some reason has been turned off. Checkpoint inhibitors enable checkpoints to be inhibited. In other words, the immune system carries on and does what it. But checkpoint inhibitors are in place because they're also there to protect your immune system from attacking you, autoimmunity. And so when you disable a safety check in your immune system, you enable toxicity. And so what's happening with not a lot of people, but 20% of people on a single agent and up to 50% of people on 2 agents, is that this autoimmune toxicity can be life-threatening, if not lethal. And so finessing that, what we're doing is we're manipulating the tumor microenvironment to make it more immunogenic with less aggression, with less systemic, an affront on the system. So yeah, nanomedicine for everything, literally. And nanomedicine nanotechnology is a very big term. Nanorods, nanoparticles, nano encapsulators. There's all kinds of nano. This is what we love about the gold, is that it is a therapeutic device in and of itself that you can action, and B, it's a payload. It's a vehicle to carry additional things to that coalface, to that thing that you're trying to perturb. And so it's exciting for us. It's one of the grails. I'll be interested to see what the world brings us, because this is stimulating everybody's imagination, as you can well imagine.
They say when all you have is a hammer, everything looks like a nail. Instead of making a bigger hammer, it seems like you're making the nail is easier to see.
Exactly. Wow, that's
right.
Part. Put that on a shirt. That's what we're doing. Exactly right.
That's it. Look, one last question here. What about bone tumors? Is that on the horizon?
Great question. And in fact, it is on our horizon as well. We're looking at it particularly from bone mets. There are primary bone tumors, cancer, sarcomas, and many cancers spread to bone. And it turns out that near infrared light passes very well through bone. So we can infiltrate a bone-bearing tumor, either primary or metastatic, with the gold nanoparticles, either directly or indirectly. So this is one of these things where we'll be looking at those conjugates. So that the gold nanoparticle has a pathfinder, a GPS on it to get it to where the problem is in a bone, for instance. And there's lots of ways to do that. And then when it's there, we can activate it because we know where the tumor is in the bone, and so we can activate it quite easily with our near infrared light. So yes, the long answer to that question was yes, it's targeting bone.
Excellent. Well, look, we're over an hour here, so I think we should wrap it up. Any closing comments, David, from you before we tidy things up?
No, I mean I've been following Carmen's research since the very early days, and it's truly just encouraging to see it come this far, and I can't wait to hear what you guys are up to next. Thank you for inviting me to participate today.
Thank you for joining us. Thank you, everyone. We always look forward to your questions, and we look forward to keeping you up to date through the summer as we push through this really exciting trial. You know, we, as I say, we'll have results. We aim to have results by the end of the summer. We're happy with enrollment, and we'll keep folks apprised. Thanks, and don't hesitate to be in touch. Check out our website or contact us individually. Thanks, everyone.